Synaptic transmission and the structure and function of neurons. So let's make a start on this then. So second bullet point of biopsychology. This is also the second time I've tried filming this because my stupid Chromebook died on me. One of the most technical bullet points that you actually need to know. So the AO1 here needs to be sharp AF. Stop playing. Shut up. So yeah, there's not really much evaluation here. What is particularly important about this bullet point is you really need to know the AO1 because it synaptic transmission comes up, I would say, at least 4 times in the spec. At least 4 times. So it comes up in addiction, comes up in schizophrenia, comes up in psychopathology, as well as biopsychology as well. So you really need to be hot on your understanding of synaptic transmission in particular. But there's 2 things we're going to be looking at today. There's going to be the structure and function of sensory, relay, and motor neurons, but there's also going to be the process of synaptic transmission, including neurotransmitters, excitation, and inhibition. Now, I'm guessing most people who are watching this are actually second years, maybe revising for a mock or the actual exam, because you do need to know this, right? You need to know everything, but some things are more important than others. As I said, it's really technical, so your AO1 needs to be quite on point for this one. Before we start, we need to actually understand what a neuron is, because it's something that you cannot see with the naked eye. None of us presumably have seen a neuron before in IRL, in real life. So a neuron is basically how the brain communicates. You have supposedly about 100 billion neurons in your head, and that's just a number to a lot of people, but there's 7.5 billion people, and there are people everywhere on 7.5 billion people on this planet. So 100 billion in your head, of which they all have multiple connections. So you're talking about trillions of connections in your brain. So neurons are actually specialized cells that transmit chemical and electrical signals to facilitate communication in the brain. Everything you do, everything you are, every memory you have, every smell you smell, every sight you process is a neuron firing in your brain. Every movement you do is a neuron firing in your brain. You're firing God knows how many neurons to pay attention to me right now, and you're doing it in certain parts of the brain. So that, as I say, about 100 billion, a neuron, so a single neuron like this, can have up to 10,000 connections to other neurons in the brain. I mean, typically you don't have that many. Let's say you might have 1,000 connections to a brain, but that's a phenomenal amount of interconnectivity. And you're required to know the 3 types of neurons, which are the following: sensory, relay, and motor. So sensory neurons are the nerve cells activated by sensory input from the environment. So for example, you have sensory neurons all over your body, sensory nerve cells all over your body. These nerve cells are connected to the neurons, and the neurons take that impulse to the brain. Think about let's not go into this too much, but think about where you have more nerve endings on your body, more sensitive parts of your body. There are multiple suggestions, which we're not going to go into, but your hands are a perfect example. Your fingertips in particular, are packed full of nerve cells. Connected to those nerve cells are sensory neurons, which take the information from the nerve cells all the way down your arm, all the way up and into your brain. And you have nerve cells as sensory neurons all over your body. Now, the main thing I want you to note down about sensory neurons is they take sensory information towards the brain. They don't take it to the muscles, they're taking it from the muscles, taking it from the nerve cells. So sensory neurons will essentially take things from the the nerve cells on the skin or on the tongue in particular. The tongue's very sensitive, or in the olfactory bulb, just at the top of your nose, to even I guess within your ears. Sensory neurons will go toward the brain. Now, relay neurons are that is core. There's two things you really need to know about relay neurons. They connect the spinal. They connect the spinal and sensory. They connect the motor and the sensory neurons together. So they're quite small relay neurons. They mainly reside in the spine, though not exclusively. And the second thing that you need to note is that relay neurons are capable of what's called the startle response. Let me give you the scenario, right? Let me give you a scenario. And for this scenario, you need to know that motor neurons, which I'm going to talk about in a second, go from the brain or the spine to the muscles, right? Imagine I'm cooking, and this is the classic example. I accidentally put my hand over a hob. Now, I don't see myself do this, right? My hand will detect extreme heat. My nerve cells connected to the sensory neurons will take that message, and they'll take it up towards, and all the while, by the way, I'm burning. All the while my hand is burning, right? It gets to the spine. Now, usually what would happen is it would have to go up to my brain. I process what's happening, come up with a response, and then I send a message down to my motor neuron to move my hand, right? Because motor is about movement. That's going to take a long time. So the second point I wanted to note about relay neurons is that they include a startle response. They can make limited movement decisions to limit damage in a potentially threatening situation. Right? So again, that's a good thing to note down. So for example, burning, and my spine essentially with the relay neurons can help me make limited movements. So in this case, I would pull my hand back, and that's called the startle response. And it's the reason why it prevents me having to put more time into it to process it and then send a message. It might just save me half a second or maybe even a second of burning. So it's an evolutionary thing. So two things you need to know about neurons. They connect the two main types of neurons, about relay neurons, they connect the two sensory and motor neurons, but they can also house the startle response as well. Now, if sensory neurons go towards the brain, motor neurons go from the brain to the muscles, and they're called motor neurons because they get you going. They help your movement much like a motor does to a vehicle. So motor neurons of the spinal cord are part of the central nervous system, and they connect to muscles, glands, and organs. So basically your motor neurons are connected to those three things, mainly muscles. That's the easy way to think about it. And if you want to use the proper term, by the way, instead of muscles, effectors, EFFECTORS, right? The motor neurons connected to the effectors, basically. These are getting you going. So, for example, if I were to twist this round, in my hands, this is all my motor neurons. My motor neurons are basically telling my fingers to move in a certain way, and I am doing that, for example. Now, my sensory neurons are also involved in this as well, obviously, to let me know where the pen is. But my motor neuron, this is how quickly it is. My sensory neurons were connecting to my brain, and then they would then kind of starting off a motor neuron response as well. Two types of motor neurons, top half of the body, bottom half of the body is the best way to think about it. But the motor neurons in your central nervous system, the brain and the spine, are known as upper motor neurons. Anything that comes out of the spine and the central nervous system is known as lower motor neurons. Now, you may have heard of motor neurons before. You probably have heard of motor neurons before. Maybe motor neuron disease. Motor neuron disease is where the long axons, So if I just skip the long axons just here, these long axons, the myelin sheaths, these little pink things here, they start to decay in people who have ALS or a motor neuron disease. The actual neuron can fire a message absolutely fine. The actual brain works fine. The problem is the message gets fired down, but because these have decayed, the message never gets to where it needs to go. And so if you're talking about motor neuron disease, if the message isn't getting to where it needs to go along the motor neurons, the muscles will never actually receive the message to move, for example. So essentially, motor neuron is some sort of like progressive paralysis, because your brain works absolutely fine, but the connections to the muscles are actually quite damaged and decayed, and therefore the muscles are waiting around like, I haven't heard from the brain in ages. But it's because the connections between the brain and the muscles are no longer working, and it gets progressively worse. So Stephen Hawking is a perfect example of this, where everything becomes slowly paralyzed. So those are the three types of neurons. The main thing you need to take away from this, though, is synaptic transmission, and this is where it went a bit wrong last time. Synaptic transmission is going to come up a lot, and so you need to know it. Best way to kind of go through this is this, right? So I have a very simple picture of a neuron, or two neurons connecting just here. So there's two phases to this. You've got the kind of the neuron-wide picture, and then what I'm going to click on in a minute is the actual synaptic transmission. So you need 8 steps to this. You need 8 things that if you want to get full marks, you need to be able to describe this in 8 stages. Now, I may get these stages wrong. I have a perfectly good slide later on, but I'm going to try slow this down a little bit in my description of synaptic transmission. Say you want to pick up a pen, right? Say you want to pick up a pen. The very basics of what happen are the following. Your soma, S-O-M-E, commonly known as the cell body, but call it the soma. Your soma needs to fire this message of picking up the pen down the axon just here. And it needs to go to several motor neurons, which is going to lead to your hand to pick up the pen. But so basically, you need to fire what is called an action potential. Now, an action potential, write this down, is an electrical impulse that contains the command from the brain, right? So it's an electrical impulse. Now, down this axon, it will fire the message electrically. But obviously, you don't have one neuron from your brain to your hand to pick up the pen. So it has to go through several neurons, basically. Now, say you achieve, and I'm going to go through this later on because this is where it gets properly complicated. I'm basically going to circle back to the start of the process with this neuron, right? See how this goes. So we fire let's say we achieve an action potential, and I will explain that when I get to this bit just here, because the cycle starts all over again. Say I fire an action potential, and I fire an electrical impulse down the axon. You have to use all this terminology, down the axon, and it reaches basically the end of the dendrite just here. Now, as you can see, if you can zoom in, that these neurons do not touch. They just about do not touch. And you can see that on, let's have a look, this just here. So the electrical message will come firing down, and it will come firing down as an electrical message here. The problem humans have is the electrical message cannot be fired across this synaptic gap. Electricity can't just jump across a gap. So what we have to do is we have to go through this whole rigmarole, this whole complicated process of converting the electrical message into a chemical message that can then be fired across. And those chemical messengers are what we call neurotransmitters. Now, there are loads of different types of neurotransmitters, each doing certain roles and jobs, for example. So dopamine is quite heavily linked to the feeling of pleasure, for example. Serotonin, they're the two big ones, by the way, serotonin and dopamine. But serotonin is linked to mood, but not just mood, learning and memory as well. And you know, that thing that we call the gut feeling. So basically, if you have a gut feeling about something, there's a lot of serotonal activity going on in your brain. So what we do is, I need to explain the structure of this as well. I'm not going to explain all of these because, quite frankly, this is working at a biology/uni level. But what I will explain are these little sacs just here. The sacs themselves are called vesicles. vesicles. They contain little beads or balls of neurotransmitter. When the nerve impulse comes down, the electrical message is converted into a neurotransmitter, a chemical message. And what happens is this neurotransmitter will start slowly moving down towards this membrane wall. And when it reaches the wall of the presynaptic membrane, the presynaptic membrane is this bulb just here. This one just here is the postsynaptic membrane. Post meaning after the synapse. This is before the synapse. What this vesicle does is it fuses into the wall of the presynaptic membrane, and it will release, like two bubbles joining together, it will release the neurotransmitters into the synapse. Okay, and remember, these neurotransmitters contain a message. In this case, pick up a pen. The neurotransmitters will go across the synapse, and they will bind or link into a corresponding receptor on the other neurotransmitter. So the message is fired across the synapse, and it enters into a corresponding receptor. So, for example, dopamine can only fit in dopamine receptors. Serotonin can only fit in serotonin receptors. GABA, which is another neurotransmitter, gamma aminobutyric acid, is to do with calming you down. That can only fit in GABA receptors. Once it does, this is where it gets a bit confusing, unless you're confused already. Once that message is passed over, the neuron is then, fired back up into the synapse, where it is basically recycled. It gets recycled up to the presynaptic membrane. Not all dopamine molecules will make it over to the side, but they will all be reuptaken back into the presynaptic membrane. Now, the message here is where it gets complicated. The message here is basically turned back from a chemical message back into a electric message or electrical message. But that doesn't mean it's actually going to fire. This is where it gets a little bit complicated. At this stage, we are at this point. We are just about back here again. Action potential has fired the electrical message down here. There's been all this chemical to and fro down here. It's been fired across the synapse, and right now we are back to this point just here. Now, what we need to establish is, does this neuron fire? Just because the message has been thrown across the synapse, it does not actually mean that the next neuron is going to fire. You need to achieve excitation in order to be able to do this. Now, I'm just going to see if I can draw you something. You're going to be very, very lucky here. I can't find you. Right, okay. Doesn't matter. Well, it does because I wanted to, but whatever. What I'm going to do is I'm going to type in action potential, and I'm going to get up a graph that looks a little bit like this. Now, what you need to know about is excitation and inhibition. on this screen just here, what you need to understand is a neuron always has a certain amount of electricity going through it. So this neuron right here has a normal amount of electricity going through it. In order for you to be able to fire, in order for that neuron to fire the next message, it needs to achieve depolarization, excitation, if you will. This is the normal hum of electricity, right? This is the normal hum of electricity. You need to achieve this amount of electricity for you to have this spike of activity. So this is where we bring in excitatory neurotransmitters and inhibitory neurotransmitters. Some neurotransmitters are excitatory, which means they often achieve reaching this threshold. Dopamine and serotonin are particularly excitatory. So if dopamine goes across, and I realize this is getting quite complicated now, but if dopamine goes across the synapse, most of the time it actually does achieve an excitatory response. GABA is an inhibitory neurotransmitter, which means most of the time when GABA is thrown across the synapse, the next neuron does not fire the message. You may question, why the bloody hell would we go through all of this process only to not... Oh, bugger, hold on a second. Why would we go through this whole process only to not fire the message? Sometimes your brain needs to calm down and not fire messages. Like if you're going to sleep, for example, your brain needs to quieten down. So what our brain does, it releases GABA in order to try calm us down. Anyway, let me just go through this. Let's imagine GABA has gone through this whole process, been fired into vesicles, fired across from the vesicles over to postsynaptic transmission. What happens is, right, normal electricity, this is GABA. Remember, doesn't fire, doesn't often reach it. What happens is GABA comes in and it goes, oh, but then what happens before it reaches the threshold? Oh, and it carries on. The message dies and the neuron carries on with a normal hum of electricity. You do not achieve depolarization, excitation, etc., etc. Now, what you're seeing here is something like dopamine. Imagine dopamine goes across the synapse just here, right? Dopamine comes in, right? The dopamine reaches the receptor just here. What happens is you get this massive shift up. You then achieve reaching the threshold. And what this is just here, what this rising of energy is the soma getting ready to fire. So right here, it's gone across, dopamine goes across. That spike that you just saw there is this neuron getting ready to fire the message, gathering energy in order to fire the message down the axon. That is that spike of energy just there. Depolarization, it's worth noting down, is the soma gathering energy to fire the message down the axon. The repolarization is it firing, and it's basically, it's almost like a throw. It's literally almost like a throw. If I give you a ball to throw, the depolarization is kind of going back, gathering energy for you to be able to fire, right? You're leaning back. The overshoot is the beginning of the throw. Repolarization is basically putting all your energy into throwing that down. Now, let me ask you this, because right down here is what you call the refractory period. The refractory period, there's many types of refractory period, which I'm not going to go into. I'll go into it in class, actually. But the refractory period is when you've, in an activity, you've spent all your energy, and for a very short period of time, you are unable to do that activity again. If you throw a ball, there's a period after you throw the ball where you are physically incapable of throwing the ball again. Right, you throw the ball, you release it, for example, and you can't then immediately throw again. Right, there's a small period where you cannot. This refractory period right here is when the neuron has basically thrown all of its energy into sending this message, and it's so spent for about a quarter of a second, or maybe an eighth of a second. It's so spent that it cannot physically actually achieve it again. Then eventually it will recover and carry on. Now, I appreciate that is actually quite complex, this whole thing. But what you've just seen here, right, with this process, is this neuron achieving an action potential and the message carrying on. It's a lot of complicated stuff, a lot of terminology we've gone through it, and I wouldn't be surprised if most of you are lost. But that is the process of synaptic transmission. You may, in order for you to actually understand this, you may have to watch this several times. Right, you may have to watch it several times. But synaptic transmission is by the process by which neurons are sent from one neuron to another. And the whole thing, which I've just described, takes about 100 milliseconds, right? 100 milliseconds. So not very long at all. Not very long at all. Why is this important? But I'm not going to go through that. A couple of, I guess, maybe a couple of bits about excitation and inhibition that you probably do need to know. Let's just see if anything's not been covered. Some neurotransmitters are excitatory and some are inhibitory. So you do need to know examples of them. So excitatory neurotransmitters such as noradrenaline and dopamine make the postsynaptic cell, so the next neuron, much more likely to fire, whereas inhibitory neurotransmitters such as GABA make them much less likely to fire. Its actual name, by the way, so if an excitatory neurotransmitter binds the postsynaptic receptors, it will cause an electrical charge, blah, 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 and the process of that likelihood of firing is called an excitatory postsynaptic potential, or an EPSP. Conversely, if you see it with inhibitory, it's IPSP, inhibitory postsynaptic potential. What these are basically the names for that process, with whether it makes it more likely to fire or less likely to fire. These are the 8 points. So you need to know these 8 points. So what I'm about to go through is a summary of what I've been talking about for the last 10 minutes. First things first, the soma fires an action potential down the axon towards the presynaptic membrane. That's that first thing, is remember, and that's an electrical charge. Second thing is the electrical impulse is then converted to a chemical impulse known as a neurotransmitter, and it starts to prepare to move towards the synapse or move across the synapse. The vesicle that contain the chemical neurotransmitters move down the presynaptic membrane and fuse into the membrane wall, where they will release the neurotransmitter into the synapse, and it gets fired across the synapse. The chemical neurotransmitter then goes across the synapse, fits into a receptor on the other side. The one thing I didn't talk about is a lock and key process. I kind of did talk about it a little bit, but where a serotonin neurotransmitter molecule can only fit into a serotonin receptor. And then stage 6 is not all of the receptors actually not all of the neurotransmitters actually find the receptors. Any neurotransmitter that remain in the synapse will be reuptaken, and that is the term actually for that hoovering up of remaining neurotransmitter molecules back up into the presynaptic membrane. If you remember OCD, the problem is that process happens too quickly. Right, I won't go into it. 7 is essentially what's going to happen now is an excitatory or an inhibitory response will occur depending on the type of neurotransmitter. And an excitatory response will increase the chances through depolarization, whilst an IPSP, an inhibitory response, will actually decrease the chances. Now, for your AL1, in a 6 marker, describing this, you need 8 points. These are the 8 points you need to be able to talk about. Okay. You've also learned quite a lot about how it works, but also the structures of synaptic transmissions, vesicles, postsynaptic membranes, so on and so forth. So in terms of questions, the basic ones you get are that bottom one. simply, A1, outline the structures and processes involved in synaptic transmission. Just outline it. This one just here is a bit more complicated because it involves you not only knowing the synaptic transmission, being able to apply it in a situation that you absolutely cannot prepare for, and you will see for the first time in the exams. So typical types of questions, as you'll notice, there's no 8 or 16 markers here. There may be 8 markers in the future, but I doubt there would be 16 marks, because how can you evaluate this process? It's very quick. There's no real research actually that you need to know. So that is it for synaptic transmission. I sense you probably will have to watch that 1 or 2 times, but I hope you found it very useful.